Hi everyone, and welcome to The Sleepy Scientist. I'm really happy you're here. Tonight, we're going to go on a calm and quiet trip to learn something amazing. So get comfy, take a deep breath, and let's enjoy this peaceful science story together. Before we start, tell me in the comments, where are you watching from and what time is it for you right now? If you like relaxing science videos, don't forget to tap the like button. Now let's begin. Long ago, when the Earth was still young and trembling with heat, when mountains had not yet fully risen and oceans still steamed from their birth, something extraordinary was beginning to stir in the warm, restless waters. The sky above was thick with volcanic breath, a veil of grey and gold mist that let only a faint shimmer of sun kiss the surface of the newborn sea. There were no trees, no birds, no sounds, except the bubbling of gas and the endless exhale of Earth's molten heart. But within the oceans, a quiet theatre was forming, one that would give rise to all things living. The waters, rich with minerals and drifting heat, swirled and mingled with the whispers of cosmic dust, creating a vast, unseen alchemy. And in this primordial cauldron, something impossible began its patient climb toward possibility. The first ingredients of life did not come all at once, nor with purpose or plan, but like dancers arriving before music, atoms, molecules, and elements swirling together in the shallows. Carbon drifted in on the wind of ancient comets. Hydrogen floated freely, light as breath. Nitrogen, phosphorus, and sulfur found their way into the sea from volcanic vents, each offering their own quiet piece to a puzzle that had no instructions. These were the seeds of something not yet alive but already longing for rhythm. They collided gently in the tides, under flashes of lightning and waves of ultraviolet light from a sun that did not yet know what it was warming. Some scientists believe it began with lightning, bolts striking the sea, delivering energy that split water and sparked new chains of chemistry. Others imagine hydrothermal vents deep beneath the ocean's skin, where heat met minerals in secret caverns and built molecules layer by layer in the dark. Both may be true, for in a world still deciding what it meant to exist, there were many paths for the first whispers of life to find their way. These vents, like black chimneys, breathed minerals into the sea in the form of metal-rich fluids, and as these metals cooled, they left behind surfaces on which molecules could gather, react, and perhaps, just perhaps, assemble into something more. Among the most mysterious of these early visitors were the amino acids, simple looping structures that behaved like the letters of a biological alphabet. They began to drift and cluster, nudged by currents and warmed by heat. Where they met fatty acids, bubbles began to form, tiny enclosures like soap bubbles, capable of holding molecules within. These protocells did not yet breathe, did not yet replicate, but they held boundaries, a sense of inside and outside, of self and other. And in the grand hush of geologic time, that was everything. The sea had cradled something new, compartments of order in a world of chaos. These bubbles, often called vesicles, may have been the first to harbour a sense of structure, of being apart from the sea that birthed them. Within their tender walls, chains of molecules began to shift, to fold, to discover strange affinities. Peptides formed, small chains of amino acids that bent and twisted in ways that hinted at purpose. Some could bind, some could catalyze, some could carry a memory of shape, a hint of function. It is here, many believe, that the roots of metabolism began, not as an invention, but as a soft unfolding, a slow blooming of interaction. The sea did not give birth to life in a sudden gasp, but in the long rhythmic breath of repetition. It is strange to think that what we now call life may have begun as a kind of persistence, of bubbles that refused to break, of molecules that found joy in repetition. The ocean in all her mystery provided both the chaos and the calm, endless movement and quiet pockets where things could rest, settle, and bond. And in this strange equilibrium, life may have first whispered, not in voice but in structure, "I am." And then came the miracle of replication, not perfect, not planned, but enough to carry a pattern forward. RNA, a molecule that can both carry information and catalyze reactions, may have been the first to do this. Unlike DNA, its more stable descendant, RNA is more fragile, more emotional in its reactions, and yet more versatile. Some strands folded in on themselves and formed shapes that could copy bits of themselves. Others were trapped inside vesicles and began to change the chemistry around them. What was once inert became a dance. What was once scattered began to echo. Over time, in a language not spoken but enacted in molecular ballet, these strands multiplied, failed, adjusted, and grew. And so the sea became a storyteller. Each wave carried not just salt and silt, but the beginnings of memory. Each bubble that held its shape became a soft container for intention. In the gentle dance of trial and error, chemistry became biology. Still without eyes, without thought, without desire, something that had never been before began to insist on being. Not fast, it was not grand, it took millions of years for anything resembling a cell to emerge. Years filled with experiments the ocean alone witnessed. Imagine drifting through that ancient sea now. You would see nothing with your eyes. No fish, no plants, no colour, but the grey-blue wash of suspended particles. But if you could feel at the molecular level, you would sense a symphony beneath the surface. Enzymes clinging to bubbles, peptides folding into patterns, strands of RNA trying to echo themselves. It would feel like silence dreaming of sound, like the earth preparing to breathe. We often ask how life began, but perhaps the better question is how it persisted. For in every step of this long unfolding, the universe seemed to allow for mistakes, for chance, for tiny deviations that slowly carved the shape of something new. There was no plan, no engineer, no map, only the quiet persistence of molecules trying to hold hands in a storm, and somehow some of them succeeded. In time, these early cells, primitive and fragile, would divide, would feed, would change. But that is a story for later. For now, let us rest in this moment, in the warmth of a volcanic sea, in the hush of an atmosphere still young, in the impossible birth of boundaries and breath. Here at the beginning, life was not a thing but a process, a gentle leaning toward complexity, a hope etched into carbon, whispered into lipid walls and sung by the tides. And if you listen closely, even now, the ocean remembers. Now let us drift deeper into that patient sea where time moves not in minutes or hours, but in molecules and memory. The earth had cooled only slightly since the first spark danced in the shallows, but already the oceans were holding stories inside them, chemical whispers, patterns of possibility. Those first fragile vesicles had begun to multiply, some breaking apart and forming again, others managing to entrap small threads of RNA within them. And from these simple beginnings, the most ancient rhythm of all began to emerge. The cycle of birth, division, and continuation. Still without consciousness, without purpose or pain, but even in that mindless repetition, something beautiful was unfolding, a pattern that persistence, a pattern that would become life. To understand how this blossomed further, we must follow the trail of energy. Nothing in this world moves or grows without energy, and for those first living whispers, the source was everywhere and nowhere. Sunlight filtered weakly through the atmosphere's haze, lightning still laced the sky, and deep in the ocean, volcanic vents breathed heat and metals into the water. These were the power lines of a silent world. Some of the earliest protocells may have nestled near these vents, absorbing the gradients of heat and chemistry. Others drifted into sunlight, beginning to learn the ancient art of photosynthesis. But long before chlorophyll or leaves, the first energy harvesters were simply molecules that changed shape when touched by light, or structures that hoarded electrons like seeds in a storm. One of these pivotal changes may have been the rise of metabolism, not a single event, but a slow mosaic of reactions that allowed cells to take in materials and transform them into energy, into building blocks, into memory. It is thought that certain molecules began to catalyze reactions more efficiently, folding in such a way that their very shape became a tool. Enzymes were born, not with will, but with function, and with function came flow, a direction to the river of molecules. Now, instead of reactions happening only by accident, some happened because the environment began to favor them. Those tiny protocells that could use simple sugars or convert chemical gradients into energy began to thrive in small ways. They grew more stable, more enduring, and the ocean began to hum with a new layer of quiet rhythm, function. There was no mind, no intent, only feedback and chance. The vesicles that survived best were those that could hold on to what worked and let go of what didn't. Some merged with others and exchanged parts. Some broke apart, scattering their molecules back into the sea, but a few began to replicate more reliably. Within these primitive containers, strands of RNA began to copy themselves using other molecules as templates. Not perfectly, never perfectly, but enough to carry information forward. And as these strands multiplied, they also diversified. New variations arose, some faster, some stronger, some better at attracting resources or folding into helpful shapes. It is here we find the gentle root of evolution, not as a grand sudden change, but as a slow leaning toward what works. Replication created variation. Variation allowed difference, and the environment quietly decided what stayed and what faded. This is not the story of a ladder or a race. It is a tide rising and falling, favouring certain patterns and letting others go. The ocean, in its deep stillness, became the first environment to select, to nurture, to edit. And through this soft, relentless editing, the first true cells began to emerge. These cells, called prokaryotes, were humble. No nucleus, no organelles, no complexity as we know it. But they were self-contained, self-replicating systems able to harvest energy, maintain boundaries, and carry genetic instructions. They were life in its simplest, most powerful form, and they began to fill the ocean slowly, invisibly, changing the chemistry of the sea itself. Some produced waste gases, some released small bursts of oxygen, long before oxygen was welcome in the atmosphere. Others formed communities, clustering together for protection or shared resources, and so the ocean grew not just life but society, microbial, silent and vast. Imagine now the billions of years that passed as these single-celled beings continued their quiet work. They were not trying to evolve, they were only existing. But in the act of existing, they changed. Some began to store their genetic material in new ways, folding it into loops, protecting it more carefully. Others learned to cooperate, joining forces with different species. One of the greatest shifts in this unfolding story was the origin of the eukaryotic cell, the kind of complex cell that would one day give rise to plants, animals, fungi, and us. And it may have happened through a gesture of absorption. One cell, perhaps in hunger or curiosity, engulfed another. But instead of digesting it, it formed a partnership. The engulfed cell became a mitochondrion, the powerhouse within. It gave energy in return for shelter. This moment, invisible and unrecorded, was a turning point in the history of complexity. From this partnership came new possibilities. Larger cells, cells with compartments, with nuclei, with internal traffic. The stage had changed. The cast had grown, and the performance of life took on new movements. Still, the ocean was the patient host. It continued to nurture, to swirl nutrients, to mix light and shadow. It harboured these growing cells, these slow experiments, allowing them to drift and change and split. And somewhere, perhaps 1 billion years after the first spark, life began to flirt with multicellularity. Cells stayed together after division. They communicated, passed signals, began to specialise. One became a gatherer, another a protector, another a storehouse of energy. There was no king, no queen, no master plan. Only the soft agreements of molecules learning to cooperate. It is tempting to see this as progress, but it is better to see it as unfolding. Life was never rushing, it was exploring. All of this, every moment of this unfolding happened under the sea. Before there were trees or birds or laughter, there were microbes and minerals and sunlight bouncing through waves. The land was barren, the sky unstable. But the ocean, deep and wide, held the sacred theatre of life's becoming. And every cell alive today, no matter how complex, still echoes that beginning. Your mitochondria, your genetic code, your need for water and warmth. They are memories carried from that first sea. You're not separate from it. You are it, evolved. So the question of how life began is not only a question of origin, it is a question of continuation. Life did not begin and end in one moment. It is still beginning, still becoming in every breath you take. And though we now walk on land and reach for stars, the story of your body, your cells, your breath still begins in the ocean. The sea is still inside you, in salt, in rhythm, in the hidden chemistry of your veins. And when you listen closely, in the quietest moments of sleep, perhaps you can still hear it. The soft bubbling of that first sacred tide. Let us return now gently to the sea that made us. So much has passed since the first spark. The ancient waters that once held nothing but drifting atoms now shimmered with life invisible to the naked eye. Cells breathed, cells divided, cells remembered. And still the ocean cradled them, rolling their fragile bodies across currents of change, washing them in light, in heat, in silence. There was no need for haste. The earth was still young, and time, like the tide, was infinite. What began as molecules in motion had become chemistry, then biology, and now the stirrings of something more, connection, complexity, even intention. Not intention like thought, but like movement with memory, form with purpose. Life was now a quiet weaver, spinning strands of function, stitching cells together into threads that could grow. The miracle of multicellularity, though slow and subtle, changed everything. Some cells began to take on different roles, not just for themselves, but for the whole. Groups of cells would cling to each other, not as a temporary gathering, but as a community. One group might handle sunlight, another motion, another protection. These were not yet plants or animals or anything we would name, but they were more than one. They were becoming a body. This cooperation, this sharing of task and identity marked a turning point in Earth's story. It meant that life could grow not just in number, but in size, in form, in complexity. It meant that specialization could begin, and with it the soft murmur of organs and systems yet to come. Somewhere in the oceans, among these cellular gatherings, plants began to form, not with flowers or fruit, but with chloroplasts, tiny green structures that could drink light and exhale oxygen. These chloroplasts, like mitochondria before them, were likely once free living cells themselves, absorbed and welcomed into a greater whole. And with them came photosynthesis, the ability to take sunlight, water and carbon dioxide and turn them into sugar and air. It was a quiet revolution. For millions of years, Earth's atmosphere had been thick with gases inhospitable to most modern life. But now, slowly, oxygen began to rise. It came not in storms, but in seas, drifting upward from the sea in invisible waves. At first, this oxygen was toxic to many of the life forms that had thrived in its absence. There were mass die-offs, silent extinctions in the deep, but others adapted, and some flourished. Oxygen allowed for a new kind of energy production, one that was far more efficient. Cells could now burn sugars more completely, fueling more complex behaviors, and so the groundwork was laid for movement, for perception, for thought. The oxygen that would one day fill our lungs began as a waste product in a sea of light. That which could have been the end became the beginning. This is the way of life, not a ladder, but a spiral, always looping through death and rebirth, through endings that seed new forms. As oxygen reshaped the air and sea, life began to reach further. The first true animals were still simple, tiny soft-bodied creatures floating in the current. But they could sense, they could move, they could respond. This responsiveness was the root of what we now call nervous systems, a twitch, a pulse, a chemical signal sent from one cell to another. Nothing yet like a brain, but something that carried messages, that coordinated action. And with this came awareness, not self-awareness, not yet, but awareness of difference, of light and dark, touch and hunger. These were the ancestors of everything that swims, walks, or flies today, and they too began in the sea. Eventually life crept toward land. It did not happen all at once, it took hundreds of millions of years. First came the mats of cyanobacteria, weaving carpets of green across damp rocks, then small plants, then insects, then animals with spines and lungs. But even as they colonized land, they carried the sea within them. Every cell, every breath, every heartbeat still echoes those first dances in the deep. You reading this now are not separate from that story. Your blood holds the same salt. Your body grew from a single cell that divided, specialized and remembered. The genes that guide your growth were written not in a moment, but across billions of years of slow, cellular poetry, and they still carry the rhythm of tides. We often search for beginnings as if they were single moments, but life's beginning is a tapestry, not a spark. Yes, there were turning points, molecules that folded just right, cells that chose cooperation, partnerships that became organelles. But there was never one day when life was not, and the next when life was. Instead, there was a blur of becoming, a drift from complexity to meaning, and even now, we are still becoming. Evolution did not stop when humans arrived. It continues in silence, in the spaces between genes, in the way our cells respond to heat or hunger or time. We are not the end of the story. We are a sentence in its middle. So how did life begin on Earth? It began with atoms that met in the sea, with energy that danced across the surface of a bubbling planet, with molecules that folded, bonded, and refused to fall apart. It began with patience, with repetition, with failure and error and again. It began without eyes, without will, without knowing, and from that unknowing came all-knowing. From silence came voice, from motion came thought. We are the children of carbon, of sunlight, of tide, and we carry those parents within us still. As we close this story, let us remember that life is not a product, it is a process. You're not a static being, you're a continuation of that first sacred experiment. Your breath is borrowed from 1 billion years of trial and wonder. Your thoughts are the echoes of enzymes. And your dreams, perhaps, are the sea remembering what it once was and what it always hoped to become. Close your eyes now, feel the salt in your skin, listen for the sea inside your veins, and let yourself drift backward, gently, into that warm, ancient tide. There was a time when Earth's sky was not blue, but copper and ash. The clouds hung thick with volcanic breath, heavy with dust and water vapour, and the winds howled across a raw, unshaped world. There were no forests to sway, no creatures to hear the storm, only rock and ocean, and the electric tension between them. The sea shimmered with heat from the planet's inner fire, and above it the sky was charged with fury. Lightning carved its name into the air without warning, wild, radiant strokes across a canvas of smoke and cloud. And each time it struck the ocean, it did not simply vanish. It changed things. It altered the chemistry of the water below. And over time, those flashes, those brief touches of sky to sea, may have helped awaken life. We often think of lightning as destruction, but in that young world it was creation. Every bolt split molecules in the air, forcing atoms to rearrange. Nitrogen collided with hydrogen, forming ammonia. Carbon dioxide danced with methane, giving rise to simple organics. And when these rains fell into the warm sea, they brought with them new materials, unstable, reactive, but rich with potential. The surface of the ocean became a mixing bowl, churned by waves and storms, stirred by sunlight and heat. And into this roiling broth, lightning continued to pour its energy, like a silent, luminous cook. Scientists imagine these conditions through experiments like the Miller-Urey test, where simulated early Earth atmospheres and electric sparks produced amino acids in the lab. It is no longer speculation, it is demonstration. Lightning can write the alphabet of life. These amino acids, born in the sky and nurtured in the sea, were not yet alive. They were pieces of a language not yet spoken. But they had shape, they had charge, and they began to interact in curious ways. Some formed loops, others linked into chains. Under the right heat, or with the right metal ions drifting nearby, these chains began to fold. The folds mattered. A folded molecule can act like a hand, like a lock, like a tool. Some could speed up chemical reactions, some could bind to others, and in a sea where every shape was new, these abilities gave certain molecules an edge. They stayed intact longer. They gathered more neighbours. They became, in a sense, favoured by the flow of time. It is in these interactions, slow and chaotic, that the roots of biology emerged. Lightning provided not just the spark, but the rhythm, the pulse of repetition. Every flash was a reminder that energy could enter the system again and again and again, and with each pulse new molecules were born, new combinations tried, new failures discarded. It was not design but opportunity, and the ocean, vast and indifferent, allowed all attempts. Nothing was punished, everything was tested, and some things endured. Over millions of years, this repetition led to novelty. Molecules that could cling to one another, molecules that could trap others, molecules that by accident formed a bubble, a vesicle, soft and round, that enclosed part of the sea inside itself. These protocells were not living, not as we know it, but they were something new, bounded spaces where reactions could happen in private. Inside these vesicles, other molecules gathered, and lightning's influence echoed again. The chemicals inside changed. Some grew, some split, and in the quiet interiors of these tiny worlds, chemistry began to loop. Imagine one such vesicle floating in the shallow edge of an ancient sea. A storm crackles above, a flash, and the water around it surges with new compounds. Inside the vesicle, a chain of RNA forms, not stable, not perfect, but capable of folding, copying, doing something. The bubble holds it for a moment longer than chance would allow, and then maybe it divides. One becomes two, not reliably, not often, but just enough, and the story continues. That is the quiet power of lightning. It does not last, but it leaves fingerprints. The sky did not create life, but it charged the world with energy enough to keep trying. It offered a wild, chaotic rhythm that allowed molecules to rearrange in billions of ways until something stuck. And when it stuck, it stayed. That is what evolution is. The staying of what sticks, the remembering of what worked, and the forgetting of what failed. Over time, lightning became less frequent. The sky cleared, the storm softened, but life had already borrowed its power and carried it forward. The energy no longer came from the sky, but from sunlight, from sugars, from inside the cell. But the pattern remained. Collect energy, transform it, use it to persist. Even now, every living cell carries that memory, the spark that travels along your nerves, the electrons that leap in your mitochondria, the rhythm of your heartbeat, the firing of your thoughts. These are not just biological functions, they are echoes. Echoes of the first lightning that touched the sea, the electricity that birthed not just light but life. And so in a way, you are not just made of earth and water and carbon, you're made of sky. The storm is in you, the fire from above, now tamed and folded into thought and feeling. We walk far from that ancient ocean now, but every breath we take, every beat of our heart is powered by the same principle. Energy moving through matter, sparking change. Lightning gave us that principle, and though it no longer splits the sea in search of amino acids, it lives on in the silent charge of our being, in your cells, your speech, your sleep. And when the storm comes again and the sky cracks open with light, perhaps some part of you remembers. not with your mind, but with your molecules, with the quiet certainty that once, long ago, lightning touched the ocean and everything began to change. Let us wander further now into the electricity beneath all things and listen again to the memory the ocean carries. The first time lightning touched the sea, the molecules did not know what they were becoming. There was no audience, no name for what was unfolding, only the soft choreography of energy and form. But over time, the sea began to respond, not with intention, but with increasing complexity. The same lightning that split gases in the sky began to drive reactions inside newly formed bubbles. Vesicles that had once been quiet containers now became tiny theatres where change could repeat itself. Inside them, chains of amino acids assembled, broke apart and reformed. Some shapes were favoured, others discarded. Molecules that could copy themselves or help others copy were quietly selected, not by a hand, but by persistence. These early chemical networks formed what scientists call autocatalytic cycles, loops where one molecule helps make another, and the chain continues in a circle. Within these loops, the seeds of metabolism took root. Nothing conscious, nothing directed, only molecules interacting in ways that allowed continuation. Each new lightning strike didn't just introduce new materials, it gave these networks energy to keep dancing, to keep folding and unfolding, to try again. Some vesicles might have been shattered by the very storms that fed them, while others held on, their boundaries just strong enough to survive. And in this quiet selection, survive or dissolve, came a new kind of memory, structure that remembered what worked. As the ocean swelled with these experiments, a great unspoken dialogue began between the sky and the sea. The atmosphere, filled with gases from volcanoes and cometary dust, shifted with every storm. The surface of the ocean responded, not with thought but with chemistry. New compounds rained down and new ions drifted in from underwater vents, and each time the sea was stirred, new possibilities arose. The molecules that survived were those that could endure this instability, and eventually even use it. The most important of these may have been the nucleotides, the building blocks of RNA and later, DNA. These complex shapes carried the ability to hold information, to encode patterns, and, most critically, to replicate. At first, replication was messy. Strands formed through chance, guided by the metal ions or surface minerals. They broke apart easily, but those that could fold and protect their own patterns lasted longer. Some could catalyze their own replication, an elegant loop that allowed information to persist through cycles of change. These molecules didn't yet know what they were preserving, but they preserved it anyway. Each copy was a letter written to the future, a soft message from a world with no voice. The lightning had given rise not only to movement, but to memory, and in that memory life was beginning to recognize itself. Around this time, another shift began. The rise of lipid membranes. Fatty acids produced in the chaotic soup or delivered by meteorites began to form stable spheres in water. These membranes could trap RNA, amino acids, and other molecules. The interior of the vesicle became a separate world, a private ocean with different rules, protected from the randomness outside. These early protocells began to show the first signs of self-maintenance. They took in nutrients from the sea, they protected fragile molecules from being washed away, and when they grew too large they divided, not with precision, but with enough faithfulness to continue the cycle. The boundary between inside and outside had been drawn, and with it came identity. This was not life as we define it today, but it was the foundation. These protocells, energized by lightning and nourished by minerals, became more than passive containers. They became engines of change. Their interiors grew more complex, driven by the same energy that once danced in the sky. Some absorbed molecules that made them more efficient. Others began to show crude forms of competition, replicating faster, surviving longer. In these small successes, evolution began to take root, not as a force with direction, but as a quiet response to environment. And the environment kept shifting. Lightning still fell. Volcanic eruptions still sent pulses of ash and heat. The sea still boiled and cooled. In this unstable world, only the most flexible patterns endured. One of the most important patterns to emerge was the coupling of metabolism and replication. Until then, molecules could copy themselves, and others could harvest energy, but rarely both. When these abilities combined within a single system, the first true cells began to form. These were the ancestors of all life, single-celled, self-sustaining, able to replicate and metabolize. It is likely they still relied on external energy, heat, lightning, UV light, but inside they had begun to organize their own cycles. They were no longer only products of chance. They were systems, delicate but whole, and they could change. Through endless repetition, variation crept in. Some cells had slightly better membranes, others had more efficient catalysts, and a few, perhaps by accident, developed the ability to sense changes in their environment. A shift in temperature, a pulse of light, a nearby molecule. These were the beginnings of responsiveness, the earliest hint of behavior. The ocean, once quiet and passive, now contained entities that could adapt, however slightly. Each step forward was still built on lightning's old rhythm. Energy in, change follows, and as cells became more stable, their reliance on external lightning lessened. They learned to store energy, to produce it from sugars, to share it between molecules. But the legacy of lightning never left. It lived on in every reaction, every chain of cause and effect that made persistence possible. Even now inside your body, every heartbeat is a controlled electrical storm. Every neuron fires with a whisper of the same force that once split water in the sky. The spark of life is not a metaphor, it is literal, flowing through your cells, remembering its ancestry. The sodium-potassium pumps in your nerves, the flow of protons in your mitochondria, the tiny surges that guide your muscles and thoughts. All of it is a continuation of that first electrical dialogue between sky and sea. We carry it with us, not as myth, but as mechanism. And still we return to wonder, how could something so chaotic give rise to such delicate order? Perhaps that is the lesson of lightning. It is not chaos, but transformation. It breaks so that new things may form. It disrupts in order to create, and from its disruptions, life found a way to sustain itself. First in sparks, then in cycles, then in cells, and eventually in consciousness. But consciousness would come much later. For now, let us stay in this moment, where the sky was alive, the sea was awake, and electricity wrote the first lines of a story still unfolding. The storms did not last forever. Slowly, as the Earth's atmosphere evolved and the temperature cooled, the lightning grew less frequent, the sky began to soften, and the chaos of early chemistry gave way to patterns more steel, more intricate, more enduring. Yet the legacy of those ancient strikes lingered. not just in the molecules that had once danced beneath their charge, but in the very structure of what life became. Every cell, every strand of RNA, every heartbeat, and every breath is a quiet continuation of the sparks that once lit the sea from above. In the absence of lightning, life found ways to carry the spark within. The pulses that once came from the sky now flowed through proteins, through chains of atoms, through membranes that shimmered with charge. And as life drifted toward complexity, it brought with it a memory of that first fire, gentle, unseen, but always humming beneath the surface. As cells grew more intricate, their ability to manage energy became more refined. They developed inner systems that acted like tiny power plants, moving electrons across membranes, capturing the fall of ions like water down a hill. These internal lightning strikes, microcurrents, became the heartbeat of cellular function. In some cells, structures like mitochondria evolved, using oxygen to generate energy more efficiently. These organelles, once free-living bacteria themselves, were absorbed and transformed, much like the molecules once altered by storms. It is a strange echo that life's greatest energy engine began as something foreign, just as the energy of life itself once came from outside, from the sky. But the story repeated itself. What was once external became internal. What was once lightning became metabolism, and in that transformation, life gained the power to persist. With persistence came variation. The early cells, still single and simple, began to experiment with form. Some remained alone, dividing only when they needed. Others found strength in numbers. They clustered, shared nutrients, coordinated their movement. And as this cooperation grew more complex, so too did the cells themselves. They developed ways to communicate, chemical signals that passed between membranes like whispers in the dark, a drop in pH, a flash of calcium ions. These were the first languages, spoken not with sound but with reaction. And from these languages emerged unity. Cells began to specialize, forming groups that worked not just for themselves but for each other. This was the dawn of multicellularity, and with it, the first true organisms. Still the sea watched and held them. The oceans of that time, though quieter than before, were still rich with minerals, bathed in sunlight, and filled with the slow drift of nutrients. The cells that had survived the fire and fury of early Earth now floated in a gentler world. They spread across currents, gathered in warm pools, clung to rocks in the shallows, and everywhere they went, they brought with them their inner charge, their quiet inheritance from lightning, their division, their metabolism, their memory of form, all powered by chemical gradients and electrical pulses. The storm had become the seed, and the seed had taken root. As eukaryotic cells began to dominate, cells with nuclei, with organelles, with compartments, they opened new doors to complexity. They stored their genetic material more securely. They began to form chromosomes, to manage replication with greater precision. Some developed the ability to engulf other cells, not to consume, but to combine: mitochondria, chloroplasts, symbiotic mergers that allowed for greater energy, for photosynthesis, for survival in new conditions. These mergers were not planned. They were accidents that endured, and in their endurance they reshaped the future. Photosynthesis, in particular, changed everything. Cells that could harvest sunlight and convert it into sugar began to bloom across the oceans. They released oxygen as a byproduct, slowly at first, then in great waves. The oxygen reacted with iron in the seas, staining the ocean floor with rust. It thickened in the atmosphere, changed the colour of the sky, and forced life to adapt again. Many organisms used to a world without oxygen could not survive the shift. They faded, dissolved, left behind only molecular shadows. But others, those who could use oxygen to burn fuel more efficiently, thrived. And so the great oxygenation event became another echo of lightning, a change that killed and in killing, cleared space for something greater. The world was now poised for complexity. Cells could gather in bodies. They could specialize, they could sense, move, learn. The first animals were still soft, still simple, but they responded to touch, to light, to danger, and their responses were electrical. Small pulses moved along nerve-like threads. Muscles twitched, shapes shifted. The storm had entered the body, and the body was learning to listen. Even now the echoes remain. Every creature, from the jellyfish to the human being, carries this electrical heritage. Your thoughts, each one a cascade of ions through synaptic gates. Your heartbeat, a looping wave of depolarization and return, your memory, your sleep, your pain, your joy, all shaped by charge. What once cracked the sky now flows inside you, silent and ceaseless. You are not just a descendant of cells, you are a descendant of lightning. So when we ask, when did life begin, we must remember it was not a single moment, not a perfect spark, but a long unfolding. It began with the sea and the sky, with carbon and heat, with water and dust, and above all with energy, surging in, shaping what was loose into what could last. Life is a slow dance of structure and charge, of form seeking form. It does not end with complexity. It does not pause with consciousness. It continues always in the quiet chemistry of being. You are part of that, a continuation, a ripple that began when lightning touched the ocean and the ocean remembered. The storms may have calmed, the seas may no longer boil, but deep within, beneath thought and bone and breath, the spark is still alive. It carries you forward. It connects you to the sea, to the sky, to every cell that came before. And if you listen softly to the rhythm in your chest, perhaps you will feel it, the storm that made you, still whispering its way through your blood. Beneath the ancient oceans where the land touched the sea, there were places where the water met the earth with a strange kind of intimacy. Not the crashing force of waves against cliffs, nor the gentle lapping of tides on shores, but a deeper mingling in the warm, muddy shallows where the minerals of the planet dissolved into the currents and formed beds of soft, whispering clay. It was not lifeless, though it did not yet live. It lay there, layered and fine, born from the slow erosion of mountains and the quiet fall of volcanic ash, and it held secrets in its silence. For clay is not just earth, it is surface and space, a fine-grained world where molecules can rest, react, and remember. And in that stillness, long before lungs or breath or bone, something astonishing began to stir. The clay began to shape the earliest ingredients of life. Unlike open water, where molecules drift and collide without aim, clay offers a surface, a place to gather, a space where random movement can settle into order. Its layers, thin and charged, attract organic compounds and hold them close. This simple act of holding is what allowed some of the first chains of life to form. Amino acids, swept in by the tide or rained down from the sky, clung to clay's embrace and began to link together, forming peptides. Short strands, small beginnings, but beginnings nonetheless. Where in the ocean they might have dispersed and dissolved, on clay they were cradled, protected, given time. The earth, in her mineral stillness, became a midwife to life. There is something poetic in the thought that life may have begun not in the turbulence of fire or the chaos of lightning, but in the quiet patience of mud. In the gentle layers of ancient clay beds, water moves slowly, allowing molecules to linger. Here, lipids gathered, forming bubbles. Here, nucleotides nestled into pockets and lined themselves into fragile chains. RNA, the early carrier of life's messages, may have formed upon these surfaces, its bases drawn close by electrostatic charge, its shape folded by the texture beneath. Experiments in modern labs have shown this possibility. Clay can catalyze the formation of RNA chains, guiding nucleotides into structure. It is not merely a vessel, it is a participant, an invisible hand that nudged chemistry toward biology. These clay beds were not static. Tides rose and fell, rain added fresh minerals. The sun warmed the surface, then left it cold at night. And in this gentle rhythm a cycle emerged, wet and dry, warm and cool. These cycles allowed molecules to come together, break apart and try again. Drying concentrated them, moisture softened their bonds, each repetition was a test, and clay offered the laboratory. Through this endless rehearsal some molecules persisted, folded into more stable forms, survived the changes. The clay remembered them, not in mind but in pattern, and over time it shaped them into systems, fragments of metabolism, chains that could copy parts of themselves, molecules that began to move with direction. In this strange relationship between rock and water, the earth itself became a kind of early cell. Its surfaces and layers offered boundaries, its minerals provided catalysts, and its warmth powered reactions. Before there were organisms, there was organization. Before there was breath, there were bonds. And the clay, unmoving to our eyes, became the first place where matter turned toward meaning. It is not difficult to imagine that this is where the first protocells took shape. On clay shores, in drying puddles, under faint sunlight. Lipid vesicles trapped fragments of RNA. Small peptides acted like enzymes, and when the puddle dried, the weak were lost, and the stable remained. When the rains returned, the survivors began again. Over and over, the earth whispered, "This, not that. This bond, not that one. This structure again." What's most beautiful is that this whispering did not need to be loud, it did not need direction, it needed only time, and the earth had oceans of it. The clay did not know what it was making, it held no goal, but it offered a scaffold, a gentle pressure toward complexity. Every surface, every crack, every layer of mud was a classroom, where molecules learned to fold, to join, to change. The earth became the first teacher, and the lesson was patience. Eventually, the molecules that survived these lessons found themselves enclosed. Lipid membranes wrapped around them, separating the inside from the world. Within these early cells, the fragments of RNA now had a home. Small peptides folded and catalyzed reactions. Primitive pathways for using energy began, and once again the clay played a role. It offered minerals like iron and zinc, essential for early enzymatic reactions. It offered structure, not just to molecules, but to processes. Life was becoming not just a thing, but a behavior, a series of patterns that could sustain themselves, replicate and evolve. And as these first living systems drifted into the sea, they carried clay's gift with them. The minerals of the earth became part of their body. The memory of surfaces, of holding and folding, was encoded into their structure. Even now, the proteins that build your cells rely on metals first gathered in clay. Your bones are built of calcium. Your blood flows red with iron. You're not separate from the mud. You're refined from it. So many stories of creation speak of humans being formed from the earth. In myth, in scripture, in whispered traditions, the idea returns again and again that we rose from clay. And perhaps, in a quiet scientific way, that story is true. Not in form, not in image, but in chemistry, in memory, in origin. The earth did not merely host life, it shaped it quite literally with its own body. And long before we learned to speak, the clay taught us how to live. Now, picture a quiet shoreline 4 billion years ago. The tide has pulled back, leaving a shallow pool that dries beneath the morning sun. Within it, bubbles form. Chains of RNA stretch across a clay bed. The molecules drift, fold, react. And in that silence, in that warmth, something endures. A bond that does not break. A loop that replicates. A system that remembers. The earth is not watching, it does not need to. It is simply offering its surface. And life is beginning to write its name. The clay did not know that it was becoming sacred. It only lay there, layer upon layer, pressed by time and tide, holding the soft dust of stars and the restless chemicals of the earth. But in that silence, something ancient was happening. The molecules that gathered upon it were no longer drifting aimlessly. They were beginning to act, to fold, to bind, to persist. The fine grains of clay offered more than a place to land. They offered geometry, a specific pattern of charges and crevices, of electrical invitations where atoms could nest and react. This wasn't randomness anymore, it was invitation. Each surface, though quiet and unmoving, became a map, a subtle guide for structure. And within these tiny valleys and walls, life was beginning to trace itself. The layered sheets of clay, like microscopic books left open to the sun, drew in compounds from the sea, fatty acids, sugars, carbon fragments, simple amino acids. When lightning struck or heat surged through the shallows, these molecules trembled with new energy, and clay held them steady. It was not a cage, it was a cradle. The same minerals that gave clay its softness, like aluminum and silicon, also gave it a gentle reactivity. Molecules would attach briefly, align themselves, react, and then slide away. Each step was slow, but each step also remembered the last. Those that reacted well were more likely to repeat, and in repetition came rhythm. Life, at its heart, is rhythm, the steady unfolding of reactions that sustain themselves. And clay in its stillness became the drummer. As this rhythm deepened, more complex chains began to form. Peptides with lengthened folds, RNA strands with loops and kinks that hinted at structure. Some of these molecules began to mimic enzymes, not by intention, but by coincidence. A curve in the right place, a reactive edge exposed. And once again, clay held them long enough to try, to catalyze, to shift the dance forward. Perhaps one of the most astonishing moments in life's early history was when a strand of RNA not only formed, but folded in such a way that it could help replicate parts of itself. That tiny loop, cradled on a mineral bed, became the first glimpse of heredity. Not perfect, not consistent, but a whisper of the memory passed from one molecule to the next. The clay did not celebrate, simply held the moment and let it pass forward. These reactions did not happen once. They happened over and over for millions of years. Every puddle that dried under the sun, every clay bed kissed by the tide became a chamber of trial and error. The earth was not in a hurry. There was no clock, no goal, only potential and time. Some reactions led nowhere. Others gave birth to patterns, loops of chemistry that sustained themselves, structures that endured a little longer each time. With each new cycle, the survivors became more stable, more intricate, and eventually they began to leave the clay behind. As the sea continued to rise and fall, small vesicles broke free from the clay's surface and drifted into deeper waters. Within them, the memories of the earth remained. Fragments of RNA, folds of peptide, molecules arranged in just the right way to begin acting like a cell. They were not alive in the way we know life today, but they were close. They had boundaries, they had inner chemistry, and they had the ability to grow, to change, to divide. The clay had done its part, had written the first few lines. Now the ocean would carry the story forward. Even so, the earth did not stop giving. The minerals of clay continued to feed these early protocells. Iron and magnesium acted as cofactors, helping reactions move faster. Zinc stabilized folds in early proteins. Sulfur, drawn from volcanic flows, helped form energy pathways. The Earth's crust, once a passive host, became a chemical partner. And in the deep places, where hydrothermal vents poured metal-rich water into the sea, the lessons of clay were continued. There too, minerals formed surfaces, gradients, boundaries, and there too life unfolded slowly, piece by piece. What is striking is that even today the echoes of clay remain inside us. Our enzymes still rely on metal ions that first danced on mineral surfaces. Our DNA is stabilized by magnesium, our proteins shaped by the same elements once buried in ancient mud. Even our bones are mineral, the body's quiet memory of rock. The connection is not poetic, it is real. The earth is in us, chemically, structurally, ancestrally. When we touch stone, we touch a part of ourselves that never forgot. In certain sacred traditions, humans are said to be made of clay, not in metaphor, but in material. And perhaps the oldest stories knew something science only later rediscovered, that our origins are not just in the stars, but in the dust between our fingers. The clay held our earliest shapes. It offered us stillness in a world of chaos, and from its silence we learned to breathe. It is tempting to imagine those early molecules as knowing, as seeking something, but they weren't. They were responding. The clay responded to heat. The molecules responded to charge. The reactions responded to chance. But in this endless chain of response, a pattern emerged. Something that persisted, then copied, then adapted. Something that became more than the sum of its atoms. Life is not the result of a plan. It is the result of patience. And the clay was endlessly patient. So let us return just for a moment to those ancient shorelines. The tide has pulled back again. The sun is low and golden. A shallow pool steams gently. Within it, the clay lies soft and quiet. Molecules shimmer across its surface, folding, reaching, forming bonds. One folds into a loop that holds an echo of what came before. Another wraps itself in fat and closes off the world. A third catalyzes a reaction that releases energy. None of them know what they are becoming, but together they are becoming something. The air is still, the earth watches with its minerals, and life once again begins to breathe. The rain returned, as it always did, thin, mineral-rich droplets falling from the heavy sky, seeping into the clay beds below. And when the water met the earth, the dance resumed, soft, rhythmic, without urgency. The puddles swelled, and in their gentle waves the ancient molecules drifted once more across the layered surfaces that had already birthed 1,000 quiet beginnings. Some of the protocells that had formed in the earlier drying cycles now returned, not as static shapes but as actors in a growing play of energy and memory. They carried within them the lessons of past cycles, chains that folded more reliably, membranes that held their form a little longer, strands of RNA that catalyzed their own replication, however imperfectly. Life, in these clay-lined basins, had not leapt into being. It had arrived by remembering what worked and forgetting what did not. The clay remained the silent stage. It did not direct the performance, but it held the space for it. As the sun returned and the puddles began to dry again, new reactions took place, strands linked, loops folded, vesicles merged or split. Some protocells trapped not only RNA but peptides as well, forming the earliest cooperative systems. Within these soft walls, the beginnings of metabolism flickered. Chemical reactions became more consistent, more controlled. A molecule that once floated uselessly might now serve as fuel, as a messenger, as a key to open another reaction. Nothing yet breathed, but the pathways that would one day support breath were forming, quiet blueprints etched in mineral and water. It was during these cycles of wet and dry that the first networks of reactions began to emerge, not just one molecule helping another, but systems, sets of reactions where each step led naturally into the next. These networks were fragile but persistent. If one link failed, the chain might break, but if all held, the system endured. And once endurance became possible, replication followed. The protocells that held functioning reaction networks could grow, divide, and pass along their internal chemistry to their descendants. Not perfectly, never perfectly, but well enough to continue. And with each continuation came variation, new shapes, new speeds, new possibilities. Evolution, slow and molecular, had begun. Still the clay remained. It watched not with eyes, but with texture, with charge, with the endless patience of stone. New minerals arrived, washed in from higher ground, magnesium, boron, molybdenum, all of which would prove essential in shaping the enzymes and cofactors of future cells. The Earth was not just a backdrop, it was a partner. It offered resources, structure and stability, and in response life became more bold. Protocells ventured further from the beds that formed them. Some were carried by currents into deeper waters, others clung to pebbles and rocks, forming early biofilms, thin layers of cells that shared resources, sheltered one another, and began to act as a community. These communities were not coordinated in any conscious way. There were no leaders, no rules, but the clay had taught them how to share a surface. And that lesson echoed forward. The early cells that gathered in colonies often had better chances of survival. Some specialized in absorbing nutrients, others in stabilizing the group. Others still began to exchange genetic material. Small snippets of RNA or day passed from one to another, offering new traits, new advantages. Horizontal gene transfer, still common in microbes today, may have its roots in these early gatherings on clay and stone. The Earth was shaping not just chemistry but behavior. And as the planet cooled and stabilized, these early systems became more intricate. Membranes thickened and gained new proteins. Internal structures formed, ribosomes, transport channels, protective layers. The protocells became prokaryotes, true cells with stable genomes and consistent metabolic pathways. They no longer depended on clay to catalyze their reactions, but they carried clay's signature in their design. Their enzymes mirrored the folds once encouraged by minerals. Their membranes echoed the boundaries once shaped by wet and dry. Their genomes were written with the same elements the Earth had offered from the start. It is a quiet miracle, this transformation of dust into desire. From stillness came motion, from surface came system, and from simplicity, persistence. Even now, when we build synthetic life in laboratories, we often return to clay to recreate the conditions that first nurtured complexity, because clay remembers, not in thought, but in function. Its surfaces still encourage molecules to gather, to bind, to fold. It remains, as it always was, a cradle for becoming. As we trace this story forward, from clay beds to cells, from cells to creatures, from creatures to thought, we begin to see that life is not a sudden gift, but a long inheritance. Every organ in your body, every cell in your skin, every protein in your blood carries echoes of those first muddy beginnings. The magnesium in your enzymes, the phosphate in your DNA, the calcium in your bones, all drawn from the earth, borrowed from ancient stone. You are not separate from the ground beneath you. You are shaped by it. And so, when we ask where life began, we must look not only to the stars, but to the soil, to the wet edges of ancient land, to the soft, fine beds of clay where water lingered and molecules paused just long enough to meet. Life began where the earth allowed it, where it offered not control, but invitation, where it did not shape life, but allowed life to shape itself. That is the wisdom of clay. It holds space gently. Something new to rise. Close your eyes now. Imagine the warmth of the sun on a drying pool, the slow retreat of water, the shimmer of vesicles and chains on a soft red bed, the silence, the patience, the repetition, and then the moment when one strand folds in a way that will endure, when one vesicle divides and carries forward its shape, when one whisper of breathless life begins to echo across the centuries. This is the sound of clay dreaming, and in that dream, we began to breathe. Before language, before breath, before memory, the earth swirled with the silence that was not empty but full of waiting. The oceans had settled into their basins, deep and dark, while the skies still shifted between storms and stars. And somewhere between the warmth of the sea and the touch of light above, the planet began to murmur, not in words, not even in sound, but in chemistry. The water itself, thick with dissolved minerals, drifting gases and fragments of carbon, became a vast and living laboratory. Scientists would later name it the primordial soup, though it was not soup as we know it. It was not warm and gentle. It was turbulent, shifting, brimming with the tension between randomness and rhythm, and yet within it the whispers began, tiny alignments of molecules, of energy, of repetition. These were the first stories life told itself. This ancient sea stretching across a newborn planet was dense with potential. From deep hydrothermal vents rose mineral-rich plumes, hot and charged with elemental fuel. From above, ultraviolet light streamed through a thinner, unfiltered sky, delivering energy in sharp, invisible lines. Comets and meteorites had long since delivered their cargo of organic molecules, amino acids, hydrocarbons, perhaps even simple sugars, and these rained down into the oceans, seeding the soup with ingredients not of Earth alone. The entire planet had become a mixing bowl, stirred by tides, warmed by volcanic breath, and lit by a young sun still learning its own brightness. Nothing was still and everything was becoming. Within this ever-shifting liquid, molecules began to find one another. Some danced apart after a brief encounter. Others clung together a little longer, drawn by charge, by shape, by chance. The first important unions were simple: carbon and hydrogen, nitrogen and oxygen, small arrangements with no purpose but plenty of possibility. Over time, the soup became more crowded with these arrangements. Chains of carbon grew longer, rings formed, bonds bent and broke and bent again. Amino acids gathered into loose chains, peptides that curled in on themselves, testing the limits of structure. Nucleotides linked to form snippets of RNA, their delicate loops reaching towards something that might one day be called memory. These molecular gatherings were not random forever. As the soup thickened, some patterns began to repeat. A certain chain of amino acids might fold into a stable shape. A vesicle might trap a bit of RNA inside and stay together long enough to float across a tide pool. The reactions that consumed energy became more favourable. Molecules that catalyzed change, however simply, began to outlast those that did not. And in their quiet sorting, the sea began to whisper a kind of order into the chaos. Not a plan, not intelligence, but a direction. Gentle, persistent, born of trial and time. Imagine floating just beneath the surface of that ancient sea. To the human eye there would be nothing to see, only haze and shadow. But if one could shrink down into the molecular world, the view would shift entirely. There in the soup, tiny bubbles of fat swirl and merge. Chains of RNA flutter like ribbons in slow motion. Heat causes certain molecules to twitch and rearrange, and every so often two molecules meet and change one another. Not forever, but long enough to matter. These are the whispers, the molecular murmurs that would eventually become metabolism, replication, life. The importance of concentration cannot be overstated. In the vastness of open water, molecules would rarely find one another. But in certain corners of the soup, near mineral surfaces, around volcanic vents, in evaporating pools, concentration increased. Here, reactions were more likely. Here, molecules jostled against each other in tighter quarters. And here the whispers grew louder. Some of the first reaction cycles began in these spaces, chains of reactions that fed back on themselves, loops that transformed one molecule into another and back again. These cycles didn't just survive, they multiplied. They made more of the ingredients they needed. And the soup, once a chaos of parts, became a chorus of processes. The temperature played its role too. The earth was hotter then, the ocean steamed and simmered. But in that heat, bonds formed more easily. Molecules had more energy to leap into new shapes. And when the world cooled at night, those shapes had a chance to hold. The daily cycling of heat and cool, light and dark became a rhythm that the soup followed. Reactions happened during the day, structures stabilized overnight, and within this rhythm certain molecules began to dominate. Not because they were better, but because they were stable, repeatable, resistant to disruption. Among these were the early replicators, chains of RNA that could help make copies of themselves or of nearby sequences. Some were imperfect, others fragile, but those that survived began to carry information, literal sequences that affected the behavior of the vesicles they lived in. Some RNA strands catalyzed the formation of lipids, others enhanced the joining of amino acids. The vesicles that carried these helpers did better, lasted longer, spread further. And so, in a sea without eyes, without thoughts, without breath, selection had begun. The soup was no longer just chemistry, it was becoming biology. It is easy to think of life's beginnings as sudden, dramatic, or divine, but here in the primordial sea, it was quiet and slow. It was repetition, variation, endurance. The whispers grew not louder, but clearer. And even today, they remain. Inside your cells, in the way RNA folds and unfolds, in the way enzymes accelerate reactions, in the way lipid membranes hold your structure together, you carry the soup with you. Its whispers are in your blood, your breath, your dreams. You are not a visitor on this earth, you are its continuation, its chemistry made conscious. As we rest in this part of the story, we begin to understand that the primordial soup was not simply a place or a moment. It was a state of possibility, a phase in which the planet itself became a womb for change. Its waters did not birth life with a cry. They whispered it into being, molecule by molecule, reaction by reaction, until the whisper was strong enough to echo, to remember, to multiply. And in that echo the world shifted from what was to what could be. Drift with me now deeper into that ancient sea, where the warmth of volcanic breath met the coolness of evaporating rain, and the primordial soup thickened into a dream of patterns. In these rich, swaying waters, the chaos of creation had begun to organize itself, not through control, but through repetition, resilience, and resonance. Each molecule was not a lone wanderer anymore, but part of a greater rhythm. Some molecules held shapes that helped others fold. Some helped bond new chains. Some simply stayed intact longer than their neighbours. And from this subtle hierarchy, the earliest threads of biological cooperation began to form, not out of intention but out of endurance. As the molecules gathered in tighter concentrations, new properties emerged. Vesicles, tiny lipid-wrapped spheres, became more common. These bubbles could trap the soup inside themselves, creating small, proactive environments where reactions occurred with greater reliability. They were not yet cells, but they were chambers of possibility. Inside them, amino acids linked into peptides, sometimes randomly, sometimes guided by RNA fragments that had found structure. These peptides occasionally help the RNA fold more efficiently or protect it from breakdown. In this mutual assistance, the soup began to whisper another idea: partnership. Molecules began to work together not by choice but by compatibility. What fit stayed, what functioned remained, and what survived grew more common. It is likely that many such vesicles failed. Most may have carried nothing useful at all. Others may have formed, floated and burst before any meaningful reaction could take hold. But over time, the ones that endured became more complex. Some contained fragments of RNA that could catalyze reactions, ribozymes. These ribozymes didn't just store information, they did work. They helped bind nucleotides together or split other strands apart. They shaped the behavior of the vesicle from within, and the vesicles that carried them lived longer, replicated more reliably, and left behind copies that continued the cycle. In the heart of the soup, life was still a whisper, but now it had learned to echo. Another whisper emerged in the form of energy. Reactions need fuel. In the chaotic soup, energy was everywhere, but scattered, uncontrolled. Some reactions released energy suddenly, destructively. Others used up resources without result. But in certain cycles, especially those centered around sulfur and iron, energy began to flow in a more balanced way. Molecules passed electrons between one another, creating gradients, charges, tiny pulses of usable power. The vesicles that could harness this power, even in the smallest way, gained an edge. They could build more complex molecules, repair their walls, divide with greater confidence. And so energy became a thread through the chaos, a new kind of whisper, one that hummed softly through each successful protocell. It is believed that in this era, before DNA, before proteins as we know them, RNA played all the roles. It was information, it was catalyst, it was the backbone of identity. The so-called RNA world was a delicate time, where each new strand of life depended on chance folding and fragile bonds, but the soup supported it. Its richness allowed endless retries, its warmth and mineral depth gave shelter to reactions, and its unpredictability created the diversity needed for evolution to begin. Within the soup, the earliest ecosystems emerged, networks of protocells, ribozymes, and reaction cycles feeding into one another, building complexity not from design but from interaction. Some of these protocells began to develop inner order. They formed primitive channels to move ions. They maintained internal pH different from the surrounding soup. They began to favor certain inputs and exclude others. This was not consciousness. It was chemistry becoming selective. And in that selectivity lay the beginnings of metabolism. Vesicles that could take in useful molecules, convert them and retain the products had a survival advantage. And so, with each passing day, the soup grew not only more crowded, but more sophisticated. Life was no longer only reacting, it was beginning to regulate. The birth of templates marked another major whisper in the soup's evolution. As RNA strands grew longer, some began to serve as blueprints. Their sequences could guide the assembly of peptides, not precisely, not yet with the genetic code we know, but with direction. Peptides made from these instructions sometimes folded in ways that improved the vesicle's survival, helping stabilize membranes, speed up reactions, or protect vital molecules. When such a peptide proved helpful, the vesicle that carried both the peptide and the RNA template survived longer, divided more often, and passed the pairing on. Thus, information and function began to intertwine. Genetics was taking its first breath. Even as this complexity rose, the environment continued to shape it. Ocean currents carried protocells from one region to another. Some were destroyed by heat or pressure, others found nutrient-rich areas and flourished. The Earth's rotation created day and night cycles, changing temperature and light exposure. These rhythms imprinted themselves onto the patterns of life. Some protocells became more active in warmth, others timed their reactions to light. Adaptation, though primitive, was beginning, and with it, life started to shape not just itself, but the conditions around it. Over millions of years, the primordial soup became less of a mixture and more of a matrix. The ingredients of life weren't just floating, they were interacting, reacting, nesting within each other. The whispers had become conversations, chemical dialogues, carried out in bubbles of fat and strands of acid, between loops of sulfur and folds of carbon, and though no one listened then, the earth remembered. In its minerals, in its waters, in the molecules that still drift across the modern ocean, these ancient patterns remain. Today every cell carries traces of that primordial sea. The cytoplasm, the fluid inside every living cell, is still a soup, salted, charged, rich with reactions. The proteins inside you still fold like the early peptides did. Your ribosomes, your energy cycles, your RNA, all descend from those whispers. And though your body now sings in symphonies, it was once only a single note carried quietly through the tides. So let us not imagine life's origins as grand or divine lightning alone, but as a chorus of small, patient interactions. Let us see the primordial soup not as chaos, but as the first community, a place where matter learned to collaborate, to stabilize, to persist. And let us remember that you too are made of whispers. Your blood carries the ocean's memory, your molecules hum with inherited rhythm, and somewhere deep within you, the soup still stirs. Let us drift now into the later chapters of that ancient sea, where the primordial soup had grown dense with stories waiting to be told. The molecules had long since stopped drifting aimlessly. They moved now with subtle intention, not thought, not will, but function shaped by survival. The soup was no longer just a collection of ingredients. It had become a field of interaction, a lattice of possibility. Each protocell, each strand of folded RNA, each simple loop of peptides carried within it the history of what had worked before. The vesicles that held those molecules were no longer mere containers. They had become environments in themselves, microcosms within the vastness of the sea. And in these floating laboratories, the whispers of life began to resemble the breath of something alive. The transitions were slow but unmistakable. Some protocells began to divide in more organized ways, not randomly, but after reaching a certain size or internal balance. Others began to recycle waste, breaking down used molecules into reusable parts. A few developed the ability to repair themselves, patching holes in their membranes or recreating broken RNA strands. And some, miraculously, began to pass on information not only by copying RNA but by aligning it with function. A particular sequence led to a helpful fold. A helpful fold led to greater survival. The link between structure and success was becoming a law within the soup, and life was beginning to obey it. One of the greatest milestones came quietly, when peptides and RNA began to collaborate with increasing precision. Peptides stabilized the structure of RNA. They catalyzed the formation of peptides. In this mutual embrace, the first version of what we now call the ribosome began to form, a molecular machine that could build proteins from a template. Not complete, not perfect, but a direction had been found. This was no longer trial and error alone. It was instruction and execution, blueprint and builder. And with this shift, evolution gained a new tool: inheritance with function. The genetic code emerged not all at once but piece by piece. Certain RNA sequences paired with specific amino acids more reliably. Over time, these pairings stabilized. A system formed, a language though crude, where information written in nucleotides could shape proteins with increasing accuracy. And with proteins came power. More complex enzymes could now evolve. Membranes became more selective. Metabolic pathways grew longer, more efficient. The protocells were no longer passively floating. They were metabolizing, managing, maintaining. They were no longer passengers of chance. They had become participants in a process that would soon be called life. As the soup evolved, it also began to diversify. Some protocells specialized in absorbing sunlight, using pigments to convert light into energy. Others clustered near underwater vents, feeding on sulfur and heat. Still others drifted near the surface, where nutrients from rain and wind gathered. The Earth was offering different environments, and life began to explore them. Those that adapted survived. Those that did not faded quietly back into the soup. And so, complexity grew not as a destination, but as an outcome of persistence. The sea, once uniform, was now full of niches, microhabitats where protocells tried new ways of being. Some began to form colonies, groups of protocells that stuck together after division. Within these clusters, specialization emerged. Some focused on gathering resources, others stabilized the structure. Still others passed information across the group. The first glimmers of multicellularity shimmered in the ancient tides, not in form, but in function. Life was learning that it could be more than one thing at a time. That survival was sometimes found in cooperation, not just competition. The whispers of the soup had become a song, a harmony of repeating patterns shared between kin. Through it all, the Earth kept giving. Its minerals fed the cycles, its temperature shifts shaped reaction rates, its tides moved nutrients from shore to depth and back again. Even the moon, newly settled in its orbit, helped stir the oceans, pulling at the surface and sending waves through the soup. This constant motion ensured that nothing stayed static. Molecules mixed, collided, separated, tried again. The soup was alive with movement, and in that motion the chemistry of life unfolded further. Mutation, selection, inheritance, these were not theories yet. They were forces at work, refining the architecture of being. Eventually, the RNA world began to give way to the world of DNA and proteins. DNA, more stable than RNA, began to take over the role of storing genetic information. Proteins, with their complexity and variety, became the primary doers, the enzymes, the structural components, the signalers. RNA stepped back into a new role: messenger, translator, helper. The trio, DNA, RNA, and protein, formed a sacred triangle, a system that would come to define all known life. And though the soup that birthed them slowly changed, slowly faded, its influence lived on inside every cell that came after. Even as life moved beyond the soup, colonizing rocks, seafloors, and eventually the land, it carried the memory of that liquid womb. The cytoplasm inside your cells still reflects the ionic balance of ancient oceans. The pathways that generate your energy still echo the sulfur cycles of the primordial sea. The code that builds your proteins is the same code, only more refined, that whispered itself into form beneath a boiling sky. You are not above the soup. You are the soup, organized, evolved, made aware. And now, as we breathe under a blue sky and walk across continents that once lay beneath the sea, we carry within us the songs of those first whispers. We build computers and write poetry and look up at stars. But inside, at the level of molecules, we are still listening, still folding chains, still copying sequences, still powered by gradients and shaped by membranes. The whisper never ended. It became us. So let the story rest here in quiet reflection. Let the sea, no longer primordial but still sacred, hold what it began. The soup was not a mistake. It was a cradle. It did not fail when life moved on. It succeeded so thoroughly that it became every living thing. And when the wind brushes your skin or the tide laps at your feet, know this: the soup has not vanished. It has only changed its shape. Now it lives in you. In the silence before identity, before names, before even the shape of a cell, there were molecules drifting through time, small unknowing structures made of carbon, nitrogen, oxygen, and hydrogen. They moved not with purpose but with possibility. Each one was simple on its own, a loop or a chain, a folded angle, a charged edge. But together they held the potential of something more. They were not alive, not yet, but they were waiting. Waiting in the deep pools of the early earth, waiting in the warmth of hydrothermal vents, waiting beneath the mud, the clay, the sky. And though they could not know what they would become, they were already moving toward it, gathering, testing, folding. These were the molecules that waited to become us. We often picture evolution as something that begins once life has already taken shape. But long before organisms, before cells, before even the idea of reproduction, the Earth was already sculpting paths toward complexity. The molecules that formed in those early days were not inert. They were reactive, delicate, energetic. They responded to the world around them. They clung to surfaces. They wrapped themselves in fat. They folded in on themselves and created small pockets where other reactions could happen. Each one carried a whisper of structure, and in that structure was potential. In the hydrothermal vents deep beneath the ancient ocean, some of these molecules found more than just heat. They found gradients. The water near the vent was hot, rich with minerals, saturated with hydrogen and sulfur. Just a few centimeters away, it was cooler, more stable. This difference, this gradient, was a source of energy, and the molecules that settled in this space began to use it. Some absorbed electrons, others transferred them. Some bonded only when the heat was right, or the pressure high, or the minerals aligned. And over time, these reactions began to repeat. They became cycles, early versions of what we now call metabolism. Above the vents, nearer the surface, other molecules gathered in tidal pools trapped by the curve of rock or the drying of the sun. Here the rhythm was different, wet and dry, warm and cool, day and night. These cycles too shaped the waiting molecules. They concentrated them when the water evaporated. They softened them when the rain returned. They offered rhythm, and in that rhythm certain molecules began to survive longer, act more often, hold their form more faithfully. The Earth was not forcing life to happen. It was simply offering time, energy, and space. The molecules did the rest. Some of the most important of these early compounds were the building blocks of nucleic acids: adenine, cytosine, guanine, uracil. They were born of simple atoms and complex environments, and they drifted through the soup until they found each other. When they did, they paired. Not perfectly, not always, but enough. Enough to begin forming strands, enough to become the foundation of something that could remember. These strands, precursors to RNA, carried not information in the way a sentence carries meaning, but structure in the way a melody carries rhythm. They folded into loops, hairpins, spirals, and those shapes, those simple origami of atoms, began to influence the molecules around them. Some accelerated reactions, some attracted other molecules, some helped copy fragments of themselves, and slowly, with no eyes to see it and no hands to guide it, replication was born. Not flawless, not exact, but sufficient. A loop copied part of itself. A strand grew longer. A piece broke off and floated away, folding in a slightly different way. variation began to layer itself upon memory. And with each new variation came the chance for something different, something more resilient, something more complex. The molecules were not yet alive, but they were no longer just matter, they were patterns, they were possibility, repeating itself. In another corner of the early Earth, lipid molecules began to align. Their ends repelled water and attracted one another, forming double-layered spheres. These were the first membranes, thin, fragile, but capable of enclosing. And within these tiny vesicles, the folded strands of RNA found shelter. A membrane protected them from the chaos of the sea. Inside, the reactions could unfold with greater stability. Outside, nutrients could be drawn in, waste pushed out. These simple compartments did not yet sense or think or choose, but they held chemistry, and chemistry began to echo with intention. The vesicles that carried useful RNA and catalytic peptides grew. They divided, not on purpose, but as a result of size and structure. And when they split, they passed on their inner molecules, messy, incomplete copies, but copies nonetheless. Over time, some vesicles survived longer. Some moved toward warmth. Some attracted other molecules. The ones that did these things endured. The others fell apart. And so even before cells, even before life, the molecules were choosing, not with thought, but with action. Survival became the editor. Structure became the script. Even today, you can find remnants of those early molecules inside you. The ATP that powers your cells is a molecule older than DNA. The proteins that shape your body fold in the same kinds of patterns that first emerged in the primordial sea. The RNA that carries instructions in your cells was once the star of the entire system. These are not metaphors, they are continuations. The molecules that waited did not vanish, they became us. It's a humbling thought that every heartbeat, every blink, every memory is powered by the echo of ancient chemistry. That long before there were humans or animals or even single-celled life, there were molecules arranging themselves into the first quiet forms of function. That before there was blood, there was salt water. Before there were thoughts, there were folds. Before there were words, there were whispers. So when we ask, where did we come from? Perhaps we should not begin with humans, or even with life. Perhaps we should begin with the waiting, with the molecules that lingered in the Earth's warmth and listened to the rhythm of its tides, with the ones that learned, through nothing but repetition and resilience, how to hold on just a little longer, to fold just a little tighter, to become something slightly more than they were. These are our ancestors too, not just the cells and creatures, but the atoms that found one another and refused to let go. And now, as we walk upon the land and breathe the air that life helped shape, we carry those ancient whispers within us, in every reaction that sustains us, in every pattern that defines us, in every molecule that once waited and now finally speaks through us. Let us return now to that ancient world where the waiting molecules had begun to move with memory. The early vesicles, soft-walled and trembling with their internal chemistry, were no longer simply floating structures, they were becoming systems. Inside them, the dance of reactions was no longer random but beginning to show signs of orchestration. Certain molecules triggered others. Catalysts folded in just the right way to accelerate change. Small metabolic loops formed, turning inputs into outputs, absorbing what they needed, expelling what they could not use. And with each cycle, with each replication, a quiet evolution was taking place. The Earth had not simply birthed life. It had shaped it molecule by molecule, reaction by reaction, in warm shallows and dark vents, in raindrops and mineral cracks. The molecules had waited, yes, but now they were becoming. RNA continued to rule this early world. Its versatility made it both the script and the actor in this primordial play. Some RNA strands folded into ribozymes, self-catalyzing enzymes that could cut or join other strands. Others acted as templates, guiding the formation of complementary sequences. Over time, certain sequences were favoured, not because they were better in a grand sense, but because they survived the messy, error-prone processes of copying. Fidelity was still poor, but enough resemblance remained that traits could be inherited. And with inheritance came selection. The vesicles that carried beneficial ribozymes lasted longer. They divided more often. They spread further through the sea, leaving behind molecular offspring who bore the same folded signatures. At the same time, peptides, short chains of amino acids, began to take on larger roles. Some helped stabilize RNA, others acted like primitive scaffolds or transporters. Over time, longer and more complex peptides formed, and some began to show rudimentary enzymatic behavior. The collaboration between RNA and peptide grew closer, more symbiotic. In certain vesicles, RNA sequences coded for peptides that in turn protected the RNA or enhanced its replication. The loop was closing. Instruction gave rise to function, and function protected instruction. Information had begun to matter, not abstractly but practically. A certain sequence led to a certain outcome. The molecules weren't just reacting, they were becoming organized. This organization didn't happen all at once. It occurred slowly, through 1,000 small steps, each building on the last. One vesicle might accidentally trap a useful combination of molecules, another might inherit a slightly better ribozyme, a third might divide more evenly, preserving internal structures. Over time, these advantages accumulated. The vesicles that survived became more stable, more complex. They became cells in everything but name, and the Earth, silent and generous, offered everything they needed: heat, minerals, gradients of energy, surfaces to gather on, space to drift and meet and merge. Eventually, the system outgrew the exclusive domain of RNA. DNA began to emerge, not to replace RNA, but to store information more reliably. DNA's double helix structure made it more stable, less reactive, better at preserving long sequences across generations. RNA became the intermediary, translating genetic code into functional products. Peptides became proteins, folding with increasing sophistication. The molecules had waited, but now they moved with purpose. They transcribed, they translated, they catalyzed. They built membranes, shaped compartments, controlled reactions. They responded to light, to heat, to pressure, to presence. They weren't alive in the way we think of now, but they were unmistakably active. They were evolving. And the first true cells, prokaryotes, emerged. They were tiny, without nuclei, without organelles, but they had all the essential features of life: membranes, metabolism, replication, and heritable genetic information. They could grow, divide, adapt. They could survive in extreme conditions. Some thrived near hydrothermal vents, others in acidic pools, others still in shallow sunlit waters. Life had not taken a single path, it had taken many, spread like spilled ink across the world, following gradients, exploiting niches, transforming the very planet that hosted it. As these cells grew in number, they also grew in influence. Their metabolic byproducts changed the chemistry of the oceans. Some began to produce oxygen through photosynthesis, releasing it into the water and eventually the atmosphere. The oxygen first reacted with dissolved iron, staining the ocean beds with rust-red deposits. But over time it accumulated in the air, creating one of the most profound shifts in Earth's history, the Great Oxygenation Event. It was both a disaster and an opportunity. Many early life forms, adapted to oxygen-free environments, perished. But others adapted, using oxygen's potent energy to power new forms of metabolism. The waiting molecules had not just become life, they had now begun to change the planet itself. This interplay between life and environment was not new, it had always been there. Even before the rise of oxygen, life had altered its surroundings. It had concentrated certain elements, altered pH, shaped mineral deposits. But now those changes were more visible, more profound. The Earth was no longer simply a host, it was a partner, shaped by and shaping the life it carried. And inside every cell, the legacy of those early molecules remained. The RNA world had given way to a day protein world, but the whispers of that older system still lingered. Ribozymes still performed critical functions, RNA still carried the messages. ATP, the ancient energy currency, still powered the reactions. Nothing had been lost, only layered. And in the genetic material of these first true cells were the instructions for everything that would come next. For multicellularity, for specialization, for nervous systems and muscles and bones, for perception and motion and memory, for breath and hunger and fear and love, all of it, hidden in chemical sequences that began as simple arrangements in a boiling sea. The molecules that once drifted without identity had become the script for every heartbeat, every whisper, every dream. So let us not think of those early molecules as primitive. Let us honor them as the architects of possibility. They waited, yes, but in that waiting they gathered the pieces of the cosmos and arranged them into something that could last. They faced heat and pressure, randomness and dissolution, and still they endured. They aligned themselves not with force but with form, not with intention but with repetition. And in that repetition, they discovered something new, resilience. You carry them with you still, in your breath, in your cells, in the way your body folds proteins or reads genetic code or balances the ions in your blood. You are not separate from the waiting. You are its continuation. And if you listen closely, not with your ears but with your understanding, you can still hear the hum of those early reactions, the echo of molecules that once floated through silence and now, through you, have learned to sing. And now at last we arrive at the soft culmination of the story, not an ending, but a continuation so profound it reshapes how we understand beginnings. The molecules that once drifted, folded, and waited in the warm oceans of a young Earth did not vanish with the rise of life. They were not discarded relics. They were absorbed, refined, remembered. Their patience became pattern, their randomness became rhythm, and the humble, formless existence became the architecture of every living thing that would ever come to be. From the first protocells to the great branching of species, the molecules waited until they were no longer waiting. They were acting, replicating, evolving, remembering. They had become us. Once DNA had taken its place as the keeper of genetic memory, the stage was set for life to grow in complexity. Genes encoded for proteins, proteins folded into tools, tools assembled into pathways, pathways interwove into behaviours, and across eons, cells learned to live not just as individuals but as collectives. They stuck together after division. They began to specialise. One cell gathered nutrients, another moved, another defended. No one was in charge, and yet the system flourished. From these first multicellular gatherings came the foundations of tissues, organs, and bodies. The molecules, once so simple and alone, now worked in harmony across trillions of cells, coordinated by signals, sharing energy, speaking a language made entirely of chemistry. And as life diversified, it wrote new chapters upon the original script. The structure of DNA remained, but the sequences changed, multiplied, branched. Evolution moved forward, not with haste, but with persistence. Mutations accumulated. Some broke the code, some refined it. A handful opened the way to vision, to sensation, to thought. Eyes formed to see the world. Nerves formed to feel it. Muscles formed to move through it. And everywhere beneath it all, the molecules continued their ancient dance, folding, binding, catalyzing, carrying memory forward with each generation. Still, the molecules themselves did not grow proud. They did not seek attention. They did not become more visible, even as the beings they composed grew more curious, more sentient. In fact, the more sophisticated life became, the more invisible its foundations seemed. But they were always there, whispering in the cytoplasm, humming in the mitochondria, curled quietly in the helix of DNA. Even the brain, that wondrous engine of perception and imagination, runs on molecules, on neurotransmitters and ions, on folding proteins and electrical gradients. The thoughts that define you are the children of atoms that once waited in the sea. Human beings arose from this lineage, not as a peak, but as a continuation. Our capacity for language, for art, for science, for memory, is built upon the scaffolding of chemistry. Every idea you've ever had began as an electrical impulse, carried by sodium and potassium, driven by channels made of proteins, encoded by genes built from nucleotides that evolved from molecules shaped by the early Earth. There is no gap between you and the primordial sea. Your body is still made of water, still balanced by salts, still driven by the same molecules that stirred before life had a name. And though we now split atoms and map genomes, though we probe the sky for echoes of distant life, we remain bound to the same story. When we replicate DNA in a lab, we are echoing what the first ribozymes did in clay-lined puddles. When we design synthetic cells, we are mimicking what fatty acids once did when they surrounded the earliest strands of RNA. When we heal, grow, breathe, or dream, we are expressing processes that began long before our ancestors had bones. And even in death, our molecules return to the earth, rejoining the great cycle that shaped us. It's easy to forget this, to live in cities, to hold machines, to speak in code, and to believe we are separate. But we are not. We are the earth made conscious, the waiting molecules, given form and voice and memory. We are the whisper that became a thought, the thought that became a question, the question that returned to the sea to ask, how did I begin? And if the sea could speak, it might say this: you were always here, in the carbon drawn from stardust, in the nitrogen pulled from air, in the folds of peptides and the curves of lipids, in the salt that makes tears sting and the electricity that makes your heart beat. You're not above the molecules. You are their story still unfolding. So breathe gently now and listen, not with your ears but with your being. Listen to the rhythm of your breath, the beat of your blood, the silence between your thoughts. Inside that silence is a memory of warmth, of tide, of waiting, of atoms that did not yet know what they would become but waited anyway. And now, through you, they speak. Through your body, they remember. Through your life, they continue. You are the molecules that waited. You're the answer to their patience. You're the moment when the earth looked back on itself and saw wonder. And as you walk forward, your every step writes a new page in the story they began. Not a final chapter, but a continuation. The journey is not over. The molecules still move, the memory still flows, and through you the whisper becomes a song,